Hello everyone. This is the last session for the Basics of Biology course, and today we will discuss the remaining topics that are part of the syllabus. And I'll be going over them as briefly as I can, but I've given the references to the original PPTs, and you can always go back to the PPTs and check the detailed discussion about the topics. Okay, so let's begin. So today we are going to discuss these topics, which include circulatory system, muscular system, nervous system, endocrine system, and excretory system. So we'll begin with circulatory system. So to give you a broad overview of what exactly is a circulatory system refers to the heart and the network of blood vessels that move around in the body carrying blood, nutrients, and oxygen. And this is because in order to utilize the food and the water and the air that we take in by our body cells, they need to be transported to different body cells so that they can be utilized. And that purpose is solved by the circulatory system. So the circulatory system consists of the heart, the capillaries, which involve the arteries and the veins. And so just briefly, if you have to classify, the arteries means those blood vessels that carry blood away from the heart, while the veins will be the blood vessels that carry blood back to the heart. And capillaries will be the site of exchange for the nutrients and oxygen within the cells. So from the blood to the cell, that exchange happens via the capillaries. And this system is important because it helps in tissues getting sufficient oxygen and nutrient, and along with this it also helps removing in the metabolic waste products. So there are different types of circulatory system. So they are open and closed. So open circulatory system, they generally are those are present in organism where the blood flows from lacuna, or basically from large open spaces and some sinuses. So what I mean by this, there is no defined channel by which the blood flows. The blood flows in very large open spaces, and because of this, the tissues are in direct contact with the blood, and therefore there is direct exchange of nutrients and gases. And here, the blood pressure is very low because there is nothing to stop or inhibit or restrict the blood flow. The blood pressure is very low in this system. And here, because it is an open space, the blood flow cannot be stopped. And what it means by, we'll compare this when we go to the closed circulatory system, what it means to stop the flow of blood, which basically means that you stop the reverse direction of the flow. And here, the respiratory pigment, basically the pigment that carries the blood oxygen within the blood, is dissolved in the plasma, and these do not have red blood cells, and they are found in higher invertebrates like insects and prawns. And compared to this, for closed circulatory system, the blood flows through a closed system, as you can see here. So you have the heart and then the blood. So this is the heart, and then you have these blood carrying the like oxygen and all the nutrients towards different body organs. And because blood, as we have seen, there are arteries, there are veins, and there are capillaries. So because of this restricted movement, or it's a channelized movement, the blood does not come in direct contact with the tissue. And because of the very same reason, the blood is blood has very high pressure in the system. And here the respiratory pigment is the red blood cells, and they're called the hemoglobin. And the other thing is, why is this more efficient? Because here the volume of the blood can be regulated by contraction and relaxation. So you have how much of amount of blood to be pumped is regulated by the contraction of blood vessels and muscles, basically the heart. And because the blood is not in direct contact with the body organs, the exchange happens at the just a sec. Just give me 2 minutes. Yeah. So yeah, Um, So to answer the question that you've asked, I that it's definitely enough to read these no lecture notes that covers all the video lectures. But in case you don't understand anything, you have these problem solving sessions. So what we try to do is we cover most of the syllabus parts that we have like that is generally discussed in the lecture. So if you read that, that should be enough for your exams. Yeah, and these kind of circulatory system, they're generally found in mollusks and echinoderms and in the vertebrates. Another added feature to closed circulatory system is the presence of lymphatic system, which consists of lymph capillaries, and lymph ducts. So these basically carry the interstitial fluid, and this we have read about this in the immune system where they have their own like they carry the lymphocytes. So what they generally do is, so you can consider lymphs to be blood but without the RBC, and they also are responsible, so they also have capillaries, and those capillaries, they unite to form a lymphatic duct. And that you can see here in the image here, that the lymphatic duct, it's actually branching out from vein and then carrying these different interstitial fluid to connect to the vein. So basically they drain all these fluids from the interstitial regions and drain it into the vein for further carrying to the lungs. Okay. Now, during the course of evolution, the number of chambers in the hearts have evolved across the animal kingdom. So we start with the simplest and we go to the complex in the mammals and the birds. So in the fishes, you have two chambers, which are the one ventricle and one atrium. And as you can see here, that in the atrium and the ventricle, so basically in general, atrium receives the blood and ventricle pumps the blood out. So here you have atrium receiving the blood and ventricle pumping the blood out, but it is the deoxygenated blood, which means that the blood goes to the site of exchange where the oxygen is like taken in the blood, and the nutrient and the waste material, basically carbon dioxide, they are given out. This happens at the gills, and then the purified blood, which is the blood which is rich in oxygen, goes via the circulation in the body to the systemic capillaries. Here again, so imagine this, to carry oxygen at this site, they will exchange oxygen with the body's cells and take a carbon dioxide from the bodies, which are the respiratory product, and this carbon dioxide will again enter heart to move via ventricle to the lungs. So this is for fishes. The site of exchange of the deoxygenated blood and oxygenated blood is gills in fishes. So instead of lungs, that the purpose is filled by lungs in the higher organisms, the fish in fishes, it's the gills. Next you have amphibians and reptiles. So both these have three chambered heart, two atriums and ventricles. Now the difference between these two is there is a incomplete septa in the reptiles. So if you see here in amphibians, you have atrium and ventricle. Now there is a clear distinction between the left and the right side. So right will be this side. So you need to imagine yourself, your back facing the screen. So your right is this side and your left is that side. So the right generally deals with the deoxygenated blood and the left deals with the oxygenated blood. Now, the oxygenated blood are meant to be carried to the body cells. So here in the left side, the blood is received from the lung because that's a purified blood and that is supposed to go to the body organs. On the right, which is the deoxygenated blood, which means these are the respiratory products which are the waste from the body cells. So these need to be carried to lungs so that they are respired out. So these are responsible for the deoxygenated blood. Now in both of them, you have atrium separate from each other, so they will receive their respective blood, but there is a mixing of the deoxygenated and the oxygenated blood in the ventricle for amphibians, and that's why this is there's the efficiency of this heart is lesser than the reptiles. The reptiles, you can see that there is a septum here, but this septum is also not complete, because of which there is certain mixing between the two kind of bloods. But that same septum is completely divided in the mammals. So here you can see that there are four different chambers, two atriums and two ventricles on the left and the right side, very clearly divided from each other. There is no mixing of blood whatsoever. So, if you define circulatory system, they generally consist of the heart and the blood. So we'll see how the blood flows through the heart. First of all, to look at heart, we you can divide anatomically it into auricles or atrium and ventricles, and the surrounding or the layers making up the wall of the heart, it can be divided into epi, being the outermost region. The middle is called the myocardium, and then the inside, which actually faces the interior of the heart, is the endocardium. Now, the next part which is to be remembered is artery and vein. We have already discussed this. Artery is dealing with the oxygenated blood. So it takes the oxygenated blood from the heart to different parts of the body, except for pulmonary artery. Now, artery are supposed to carry the, or you should remember it as this carries the oxygenated blood, except for pulmonary artery, which carries deoxygenated blood to the lungs for purification. Similarly, for veins, these carry the impure blood from different parts of the body to the heart, except for the pulmonary vein, which carries the oxygenated blood to the heart. So these are the exceptions in these cases. So this is a general flow of blood in the heart. So we'll see how it happens. So let's start with the deoxygenated blood, which is the blood which is picked up by the blood vessels from these organs or body cells and carried to the lungs for purification. Yeah. So it starts with the body's tissues draining their blood, or the blood, different veins draining their collected blood into the vena cava. So the vena cava is the input point for the deoxygenated blood. From the vena cava, it enters in the right atrium. From there, it enters to right ventricle. So the movement is always unidirectional. It will just enter in the atrium and will move to the ventricle and then to the next channel. From the ventricle, which is the right ventricle, it moves to the pulmonary artery, which is where it will be then carried to the lungs for purification. Now that's what's happening. Now at these sites, you see these names: tricuspid valve and pulmonary semilunar valve. So these are these valves. So the role of valves is to prevent the backflow, and that is exactly what is not happening in the open circulatory system. And how does that happen? So what happens is, when the blood, because the atrium is filled with blood, it opens this valve due to pressure, and all the blood is drained into the right ventricle. As soon as the right ventricle is filled up with blood, it pushes these valves back. So here the tricuspid valve, it pushes this valve back and the valve closes. As a result, what happens is the backflow of the blood from the ventricle to the atrium is inhibited. Similarly, there is a pulmonary valve when the blood moves from right ventricle to the pulmonary artery. As soon as the blood is pushed into the artery, this valve closes, so there is no backflow of blood. Now, moving on to the left side of the heart, which deals with the oxygenated blood, these will receive the blood from lungs and then carry it to the body cells. So the blood, or the blood vessel that brings the blood from the lungs, is the pulmonary vein, and it drains the blood into the left atrium. Left atrium passes it to left ventricle, and then it passes on to the blood vessel that carries the blood to different body cells, which is the aorta. Again, you have a valve here and a valve here, which is called the aortic valve, which deals with aorta, and mitral valve, which is referring to the between the atriums and the ventricle. So you see that how well regulated this whole system is, and how it is taken care of, that there is no mixing of blood, there is no backflow of blood, and that highlights the efficiency of the vertebrate heart in pumping the blood. The next important topic about vertebrate heart is about the heartbeat. At what rate does this contraction happen? That does this whole process takes place? And that is regulated by the sinoatrial node, also called SA node, also called the pacemaker of the heart. And what they generally do is that so basically sinoatrial node is basically a nervous tissue, and it refers to it maintains the heartbeat time basically. So all these what we have discussed till now completes one heartbeat. The closing of the valves is what you hear when you put your stethoscope, the lob and dub sound. So all the process completes one heartbeat. So how frequently this heartbeat is happening, basically how frequently your heart is pumping blood into different chambers, is timed by this node. And what do they do? How does that actually happen? These cause the atria to contract, and then they signal the atrovent. So there is a node here, which is just at the beginning here, and they pass the information to then atrioventricular node, so that the next round of from the like the contraction for the atria happen, so that the blood is passed to the ventricle. And what are involved in this process? So you have the sinoatrial node, which begins the contraction, gives the information to the atrioventricular node, which then passes on the information to bundle of His. These are these fibers which are present in the ventricles region. And then at the Purkinje fibers, which finally carry to all the muscles, because all of them have to contract so that the blood can be pushed into the next chamber. So Purkinje fibers are at the end, which receive the information and start contracting. Okay. And this is something we have been discussing so far. So arteries, so you can see here. So arteries are the one that carry the oxygenated blood. When they divide into these fine tubules, they are called arterioles. Why is that? Why is it important to have such thin-walled vessels? Because that will facilitate the exchange of the oxygen, or the like oxygen on taking up the carbon dioxide. So similarly, you have veins which differentiate into venules, and capillaries are even finer blood vessels. And you can see that these are very fine vesicles here. They only have one lining, which is the endothelium. And because of this thin layer, they facilitate the exchange. And the second part of the circulatory system is blood, and they generally consist of blood plasma and the other components, which are the red blood cell, white blood cells, and the platelets. Each of them have their own function. Like, for example, red blood cells are responsible for carrying the oxygen because of the presence of hemoglobin. White blood cells are involved in the defense system, and platelets are mainly involved in this process called blood clotting. So whenever you have a wound or injury, you see that there is a formation of a clot, which is this hardened tissue, and that is facilitated by the platelets. So what do they do is, when there is an injury, so they stimulate these platelets, also called thrombocytes, and these thrombocytes are attracted to the region of injury. Once they reach there, they further release factors so that they initiate the blood clotting cascade. And the what actually happens is that you have this coagulum, which is the network of fibrin threads. So the main component of your clotting is the fibrin threads, which are facilitated by the platelets. So you can see in this diagrammatic representation that, let's say you have a damaged blood vessels here. So at first, at the site of injury, you have the movement of these platelets. So the platelets will first clog the site of injury and then release factors which will attract the fibrinogen, and this fibrinogen will come and then it will be converted to fibrin, and then they will form this mesh like structure, so that they prevent the blood flow, and then on that fibrin you will have new tissue formation, which is what happens when your injury heals. So that's why platelets are also equally important. And related to these topics, you have something called blood pressure, which is something I'm sure everybody would have heard. Blood pressure is basically the force of blood against the artery. So how quickly, or how smoothly the blood is flowing through arteries. And this is further divided into two: systolic and diastolic pressure, which is something written as, for example, 120 by 80. And this 120 refers to the systolic blood pressure and 80 refers to diastolic pressure. Now, what is systolic and diastolic? So as you can see here, systolic refers to the pressure which is exerted when the heart beats, while diastolic is the pressure exerted on the wall of arteries when the heart muscles relax in between two beats. So after contraction, when the when the heart muscles are not contracting, but rather they're in the relaxation mode, the standing pressure is called diastolic pressure. But when they are they're contracting and pumping the blood, that is called systolic blood pressure. And there's certain set of certain limit how much the blood pressure should be. So generally, because of contraction, systolic blood pressures are quite high, while the diastolic blood pressure, because it is a relaxation pressure, it is quite low. So that's why you have 120 by 180, which is 120 by 80, where you can see the 80 is the diastolic, which is lower in number, 120 is higher. And when you say hypertension or blood pressure patients, you will see them having more of these number, 150 by something, 180 by something. That is when your blood is under tremendous pressure to move through the artery. And a simple guess why could this happen is, let's say you have a tube and you are trying to push something from one end to the other. Now, inside that tube, inside the lining, you stuff it up with something which is coating the wall of the artery, and then you try to push something from it. Because of the resistance that it faces, because of the external lining, because now you imagine the pump, the pipe, the diameter has reduced further because of the deposition. The thing that we are trying to push through the artery or the tube will face more pressure, and that is what happens in patients like diabetes, where what happens is there is a formation of plaque is like basic deposition. So you have these arteries, the lumen of the artery, which is basically the diameter, the lumen of the artery reduces because of this deposition, which is called plaque. And because of now reduced lumen, the blood is flowing through extremely high pressure, and that is what is reflected in their blood pressure numbers, which is also called as hypertension. So that's the idea of why this is important. Yeah, so summarizing all of these, there are some set of questions. So we'll try to go over them. The first is like I have discussed most of it. If not, I'll come to this question and discuss as we go. So the first is the reason why tricuspid and bicuspid was are closed is like, what is the reason for them being closed if they are not under heartbeat? The type of circulatory system seen in earthworm is what? Next is so there are two words here, and which of these means same? And bundle of His is a network of, or what is a bundle of His when we talk about the heartbeats? Okay. So let's tally the answers. The first is why they are closed, because the attempt to prevent the attempted backflow of blood into atria. So the reason why they are closed is to maintain this attempted backflow, to reduce the related to the backflow. Next is the type of circulatory system that you see in earthworm is closed circulatory system. We have discussed it in the vertebrate section. And so which of these two words are same? So blood cancer and hemophilia are two different things. Hemophilia is when the blood is not clotting, and blood cancer relates to the over multiplication of the blood cells. Osteoporosis and arthritis are two different kind of diseases or problems. Arthritis is an inflammatory disease. Osteoporosis is something to do with bones. But pacemakers and SA node being same is something we have discussed when we were discussing about the heartbeats. And bundle of His is, as I mentioned, these are the muscle fibers which are present in the ventricle wall, which are at the just before the Purkinje fibers, which actually relay the information to the Purkinje fibers. Okay, so the general difference between the systolic and the diastolic pressure, we just discussed the normal systolic and diastolic pressure. So what is the difference between them? What is the correct route of a pulse making impulse stimulus? So basically, what is the route of the movement of the impulse in the heart? So when there is a clot formation, which blood cells get damaged or which blood cells are involved? And the oxygenated and deoxygenated blood in the frogs, which is the amphibians heart, they are mixed in which part of the heart? Yeah, so 120 by 80, so the difference was 40. The correct route is from SA node, sinoatrial node, to the atrioventricular node, to bundle of His, then to Purkinje fibers, and then the heart muscles for them to contract. relation platelets are related to the clot formation, and blood get mixed in the ventricles for the frogs because they have the because they don't have the septa. And the next last set of questions are this is something we have discussed, though I have not given you the term, but you can try to guess. If not, we'll get the answers in a while. So, what is the condition which is called when there is a plaque formation in the inside of arteries? And given some problems of heart and what is the associated issue with that, can you guess which one is correct? And the limbs serve to do what? And the deoxygenated blood in fishes is turned, so which is the site of purification for deoxygenated blood? So the first condition is called atherosclerosis. Arthritis, arthralgia, and arthrocentesis are not related to this. It is the atherosclerosis, which is a wrong match. So heart failure, as the name suggests, it is related to insufficient blood supply to the body. Heart attack is a related term, but it is referring to the insufficient blood supply to only cardiac muscle, only to heart. Heart failure is basically for the entire body when the heart fails to pump the blood. Angina pectoris is when you have a chest pain, but cardiac arrest is not lumen narrowing. Rather, it is again because of the functioning of the cardiac muscles. And also, this also we have discussed. Lymph is basically to return the interstitial fluid, the fluid which is in the interstitial spaces, to the blood. And the site of purification of blood for fishes are the gills. Okay, moving on to the next topic for today, which is muscular system. Again, it will be all very brief. I've tried to cover as much as possible. Okay, so muscular system basically they coordinate locomotion. Locomotion is when there is a movement, like any kind of movement, and these are coordinated by the skeletal, neural, and muscular system. And we'll discuss about muscular system. And these muscular muscles basically they originate from the mesodermal germinal layer. And if you say something to be a muscular nature, a muscle, the properties that define it is contraction. So it is able to reduce its length, extend its length, and the fibers can get excited, which we will see. So this is referring to when we say electrochemically, and elasticity is basically to expand. And so all these different muscles also can be defined on the basis of where they are found. So they can be skeletal, visceral or smooth muscle, and cardiac muscle. So these are the three kind of muscles. So first we'll discuss about skeletal system. Skeletal means bones. So the muscles which are attached to the bones, they are the skeletal muscle system. And you see them when you look at their cell shape, they are single, long and cylindrical shape. They can be striated. So striated means multiple of them being connected to each other, and that's why they are multinucleated. Skeletal muscle, because they are connected to bones, they are voluntary, so you can control them. So you can decide when to move your hand and when to move your leg. Cardiac muscles are, as you can guess, these are located in the walls of heart. By appearance they have these branching patterns, and these are also striated, so they are long running with each other, but they are uninucleated, and so that's how they separate from the skeletal muscle. But these are involuntary as well, so you have no control over how these muscles will beat, because they are under the nervous system's control, and they have their own pacemaker. Similarly, smooth muscles are also involuntary, and the smooth muscles are generally within the organs. For example, stomach lining. As you know, stomach lining also have muscles. Esophagus. These are muscles that undergo contraction to pass the food down. So these muscles are also involuntary, and these have a completely different kind of cell shape, where you have again single cells, but they are this fusiform. Fusiform means something like spindle shape, no striations, and they are uninucleated. So these are how you can classify these different muscular systems. Then so we'll discuss in detail about skeletal muscles. So you can see how a skeletal muscles are something like you can define them as connected to bones. So the connection between bones and muscles are called tendons, while the connection between a muscle and a muscle is called ligament. So when you have ligament tear, basically it means that the muscles got overused and there's a tear or there's a disruption in the fiber. But this is when the muscles are connected to the bones. So you can see that the skeletal muscles, they have all the unit of a skeletal muscle is a muscle fiber, and there are multiples of muscle fibers running in the muscles that we observe. And those two are not directly, like all of them are not running parallelly. They are first arranged in something called fascia, so a bundle, and this bundle are then held together by connective tissue. So to move from the bones to the muscles, let's do that. Let's start with here. This is the connection tendon, and then you move to this. These are the layers of the muscle tissue. So you have the epimysium again at the outside, complete outside, and then your endomysium lining the inner part. And then you have this perimysium here, which lines the bundles. What are these bundles? So let's move to one of these bundles, which is called fascicle. So you have these muscle bundles together. Each of these muscles bundle has form of these fibers. So let's count. Let's say there are 6 fibers forming one muscle bundle here, or one fascia, and the fascia is lined by the perimysium. So when you see the further of like detailed structure, you can again see here. So you have these multiple muscle fibers lined by the perimysium, each of them being called a fascicle. Take one fascicle, you will see that there are again multiple fibers. Then you take one fiber and you see that they are further formed of these myofibrils. So there are multiple myofibrils which come together to form one muscle fiber. And so within so like the fluid that is surrounding the or the membrane that is surrounding the myofibrils or muscle fibers in general is called the sarcolemma. And because of these multiple muscle fibers running like in parallel to each other, they have this multiple nucleus, and that's why so multiple nucleus situation is called syncytium. And you will realize why sarcoplasm of muscle fibers have very high amount of calcium. We will see when we do the muscle contraction what is the importance of calcium. And now if we move to myofibril, which is the unit component, we will see that they are formed of these kind of bands, light and dark band. And this is because of the presence of the filaments, the myofilaments, which are again thin and thick. So these thick and thin filament, as you can see here, if you just see, so just imagine that these are all myofibrils stacked on top of each other. When you just look from look under from the point of morphology, you will see that they are striated. So there is this light and dark banding pattern. That banding pattern is because of filaments, which are these actin, which are the thin filaments, the lighter portion. Thick filaments are the myosin. So you have actin and myosin, which are responsible for the movement of the muscle. And on top of these, you have these different other proteins, which are tropomyosin and troponin. And these tropomyosin and troponin, they will regulate the binding of actin and myosin. And other than that, you have these different proteins which are responsible for this multimeric unit. So all these different actin coming together, different myosin units coming together, they are also facilitated by different proteins. So this we have already discussed. This again says the same thing. So that same striation we can see here. Now, if you see the striations here, you see there are different kind of bands: A band, I band, and H zone, and Z disc. So if you see a sarcomere is this basic unit of muscle, the muscle fiber, where you can classify the thick and thin myofilaments boundary. Now you have this A band. If you look at these bands, these dark color band, these are the myosin, the thick filament. And if you look at these lighter green color bands here, these are the thin filament. Now you have these regions where these are overlapping. So the region which relates only to the length of the thick filament is called A band. And to the region which only relates to the thin filament without the overlapping of the thick filament is called the I band. Now, similarly, the region without the overlapping of thin filament consisting only of the thick filament is called H zone. And the lining where this, so you have this is one set. Similar pattern is present on the left and the right side. So where these two thin filaments meet, that is called the Z disc. And these are important because you will see how these distances is shortened when you have the muscle contraction. So we'll do that right now. So what we have discussed is what you find here. So this is a relaxed state where there is no contraction. So you have a certain H zone, you have a certain I zone, and then you have the band, A band, where you have the overlapping of the like thick filament and the thin filament. Now, how does the muscle contraction begin? The same way, it has to first receive a cue, and that cue is given in the form of a neurotransmitter. So you have this acetylcholine. So what happens is when there is a stimulation by the nerve or brain or the spinal cord nervous system, which is brought about by neurons, uh, the first thing is, so here is a muscle, let's say. So there is a release of acetylcholine from the synaptic vesicle. They will come and bind to the receptor and will begin something called depolarization. And then when you have acetylcholine binding to these receptor, it will subsequently open sodium channels. When you have sodium channel opening, you have sodium channel moving inside this sodium moving inside the cell. Now, like I was mentioning about electrochemical properties of the cells in the body, these all ions, they are maintained at a certain level, and any deviation from the normal concentration leads to something called action potential, because there is a charge differences now in the cell from its usual condition. So influx or an efflux can both change the potential of the cell. So here, the movement of the sodium inside the cell generates an action potential in the muscle fiber, and now as when it moves through the muscle fiber, it will bring about the changes it is supposed to bring. For example, here it will lead to muscle contraction. So now sodium has moved in and the action potential has started to move in the muscle fiber. Because sarcoplasm has calcium 2 plus ions, these, as soon as your action potential is moving across, the sarcoplasmic reticulum will get activated and they will release calcium. Now calcium is released, and now we will see how this contraction takes place. So calcium will go and bind to troponin. Troponin was originally sitting on actin, masking the myosin binding site. So you can see these thick and thin filament, right? They are separated from each other for the time being, because on the thin filament you had this tropomyosin which was sitting at the side where myosin is supposed to bind. But calcium 2 plus, now being higher in number, goes and binds to the troponin, removes troponin from the actin. Actin is now free. Myosin now comes and binds to the like actin because of the presence of tropomyosin. Now, as the myosin binds to, so you can see here, this is the point where you have the myosin binding to the actin. Myosin has a head, and this head bends. So it binds and then it pulls actin towards itself. This is not changing, but the thin filament is moving towards the what do you call the thick filament. So as this thick filament is moving, a thin filament is moving towards the thick filament. This is called power stroke, and this is where the muscle has started to contract. So we'll go back to this and we'll see what happens in contraction. Look at the I zone. This I zone is one get contracted in during the contraction, and when it is maximally contracted, the Z line, which is the intermediate line, it is completely disappeared. The I zone has also completely disappeared. All the filaments are like in continuation with each other. So this is contraction. And what happens in relaxation? These guys with, so actin and myosin will move apart from each other, and these distance will again, they will move apart from each other, and you will come back to this kind of zone. That's what happens in the contraction. So this process of them binding and detaching and binding and detaching continues till the action potential is there. As and when the signal from the acetylcholine is stopped, everything goes back to normalcy, and then this comes to complete relaxation process. But till the signal is not stopped, this binding and pulling towards itself, binding and pulling towards itself continues. So you have excessive muscle contraction taking place. Okay. So I have tried to put two slides for questions for each of these subtopics. So we'll try to answer that. So first is tendons and ligaments. We have discussed both of these terms. What are these basically, if you have to define them? Next is skeletal muscles. Yeah, I'll come to the answer. We'll put an hold to this. Second is skeletal muscle bundles, which are held together by a common connective tissue. We have discussed this. What is the layer called? And then in the striated muscles, the functional unit of contractile. So if you have this contraction, which is the functional unit, and for muscle contraction, which is the iron, which is the most essential. Yeah, so coming to the first one, it is connective tissue, yes, but it is a fibrous connective tissue because it is connecting, it is fibrous in nature, right? So they are also running parallelly to each other. So they are fibrous in nature. Second was fascia, and we had discussed this. Third is sarcomere. Fourth is sodium because it is the sodium influx into the cell that starts the action potential. Okay. So the next is the neurotransmitter which upon binding leads to the influx of sodium, so which is the most essential neurotransmitter for muscle contraction. Next is the striated appearance of skeletal muscle is due to which of these proteins. So repeating pattern of which of these proteins. So there are series of some events. So first is H zone disappearing. Second is A band widening. Third is I band shortening. Fourth is width of the A band is unaffected. And fifth is M line and Z line coming closer. So in muscle contraction, which of these events are actually taking place? And the next is the muscle tissue where you will see cross-linking between the different units. So first is acetylcholine. We have discussed this. Second is also easy, actin and myosin. Third is cardiac muscle because you see the cross-linking there. And next, the last one is which of these event takes place. So you have all of them except for the A band widening because you don't have widening because A band is not getting any change. It is the thin filament which is moving towards the thick filament. So that is what is getting shortened. Okay. Now, the next topic that we'll discuss is nervous system. Keeping it very short in lieu of time constraint. But again, if you want to discuss it in detail or read it in detail, you can always go back to the original slide. So yeah, nervous system is basically when there is any stimuli and any living organism is responding to the stimuli. So here you have this a whole system which is connected by networks of nervous tissues and neurons, and there is an organ system which is involved, which is responsible in a living organism responding to a stimuli. So for example, let's say this is water and you want to have water. So you see a water. So first thing is the receptor is the eye. There's a sensory neuron which sends the muscle, so the message to the brain. The brain says, okay, you want to drink water, so you need to first pick the glass of water. So it will send a message to the motor neuron saying that, okay, pick the glass up and drink it. So that information goes to the muscle and you start moving your hand so that you can pick the glass. This is one example. The other is very classic example where you have a source of heat, let's say a candle, and you touch it. As soon as you touch it, you have so this sensory neuron sends a message to the spinal cord. The spinal cord then sends message to the efferent neuron, so effector neuron. It goes to the muscle and says, remove your hand from the source of the heat. So you remove your hand as a very quickly, and this happens to you, right? You touch something on it and remove it very quickly. So you can imagine how quickly these nervous system function for response to a stimuli. So you just saw that there are two message receiving center, the brain and the spinal cord, and these are the message releasing center for any nervous system. So, like I mentioned, so the nervous system can be divided into several classification, like peripheral and central. Peripheral, so these are the components, or where are the nerves basically present. So central means the one which are controlling the whole system. So that consists of this brain and the spinal cord. Peripheral have all the nerves in the body because they are the one that are carrying out all the information, all the activity. In peripheral, you have two types again, autonomic and somatic. Somatic is something that you can control, which is in your skins and your muscles, which are in the periphery, so you can control them. But autonomous are in the organs, so in the internal organs and glands where you cannot control them. So that is autonomous. Autonomous are further divided into sympathetic and parasympathetic, and we'll see what they mean when we come to that. So let's start with central nervous system, which consists of the brain and the spinal cord. uh so brain, let's start with brain. So if you see brain, these are the different, so these are different views of brain. Like if you view brain from a lateral means side. If you see it from side, you will see it like this. Sagittal means, if you in the lateral view, if you divide it in from the middle, how it will look inside. Superior means when you're looking it from the top, and inferior means when you're looking it from the bottom. But all of these point to the same structure, that you have a cerebellum, you have pons, midbrains, and then you have the forebrain. So there are mainly, you can divide them into forebrain, midbrain, and hindbrain. Other than that, the important factor, the important characteristic of brain is that it is enclosed in the skull. So this is a very delicate like delicate organ, so it is protected by several encapsulations. So skull being most important, which is covering from the frontal, lateral means side, and dorsal region, that is the upper portion. And other than that, the this tissue, this organ is covered by triple membrane of connective tissue, which are called meninges. And the three layers are durameter, arachnoid, and pyometer. Durameter being the outermost, and because it's the outermost, it's very strong, and it is non-elastic, so it's very like it's because it's made of a collagen that is very strong in nature. Next, the middle portion is called arachnoid, which is again very highly folded. So and then the last one is pyometer, which is the innermost, and it is thin and delicate and very vascular. What vascular? Because they have to be connected to the brains with the, so for exchange of nutrients and all the information. So that is connected to the brain, and all of these will have some fluid which are, like present in between them. So for example, here durameter and arachnoid, you have a subdural layer. Arachnoid and pyometer, you have a subarachnoid layer. So all of these are highly convoluted and very detailed, like very nicely structured. And for as I was mentioning, mainly you can divide brain into forebrain, then the midbrain. So the forebrain being the this region, main region, and that will consist of cerebrum, thalamus, and hypothalamus. Mainly cerebrum again, and then thalamus and hypothalamus. Then you move to the midbrain, which consists of tactum and tegmentum, and then hindbrain, which consists of pons, medulla oblongata, and the cerebellum, and then which continues into the spinal cord. So forebrain is where the maximal information is retained by the brain. They have multiple lobes. So you have this frontal lobe in the frontal region. Then you have the temporal lobe. Then you have the parietal lobe and occipital lobe. And you can see in this image that all of these different region. So this portion of cerebrum is called cerebral cortex, and they have these different regions which control different activities. So let's start with few of them. So you have primary motor cortex, which motor being voluntary movement, and then you have premotor cortex, which is working with the primary motor to carry out the movements of voluntary actions. And then you have prefrontal association areas. The name says it is association. So you're perceiving something associated with something, and then you are acting out. What does it mean? So you have idea and plan for voluntary movements, and again thought personality. So you see what kind of personality you are, what kind of thing you are thinking, what kind of ideas you are planning is also controlled by a certain portion in the brain. Then you have this Broca's area, which is responsible for speech. Olfactory, as a name suggests, it is for smell. Limbic is for emotions, learning, and memory, one of the main functions of brain. And then you have auditory cortex, which is referring to hearing, as the name suggests. Again, you have different other regions for language comprehension, for vision, and for other somatostatic information, sensory information. So major portion of the forebrain is controlled by cerebrum. The other part of forebrains are the diencephalon and thalamus, which are mainly responsible for getting the sensory impulses and then relaying the information to cerebral cortexes. So you have these, so they will have different points that where they can like collect the information and then they pass it on to the cerebral cortex, which again then functions in the appropriate region of this cortex and gives out the necessary information. And then you also have hypothalamus, epithalamus, and metathalamus. So you have just read thalamus. In nearby in the region surrounding thalamus, you have hypothalamus, epithalamus, and metathalamus, each with their own function. Hypothalamus is the master gland, because when we will do endocrine system, we'll see that all the hormones which are responsible for maintaining the hemostasis in the body is secreted by hypothalamus, or is stimulated by secretions of hypothalamus. Epithalamus has pineal gland, and which is again very important for the body. And then metathalamus, they are generally responsible for hearing and vision, so they also have very particular function. Moving on to midbrain, as the name, as you can guess, this is a connection between the hindbrain and the forebrain, and they also have their own set of nerves which are involved in the trochlear being hearing and oculomotor being visual. So visual and auditory information are also processed in the midbrain, and with that they can regulate the eye and the eyelid movement. And they're generally so if you have to see what are they made up of, they are made up of tactum, they're made up of tegmentum and the cerebral peduncles. So, and they also have this aqueduct here. So the main function of midbrain would be to regulate auditory and visual movement, along with some emotion like pain or any mood or any breathing also are partly regulated by the midbrain. Last section is hindbrain, which consists of pons and the medulla and cerebellum. So pons are generally controlling the sleep-wake cycle and the dreams, and the cerebellum is responsible for the muscle movement. So if the muscle movements are generally controlled by cerebellum, and this, because it is important in muscle movements and motor skills, they are responsible for activities like swimming and cycling. And medulla is responsible for all the internal organs, for example the stomach contractions or the cardiovascular movements or anything which is to be doing with the involuntary muscles. Moving on to spinal cord, again spinal cord also have their own what do you say importance. So they have their, so the most important thing about spinal cord is the this long channel running so this runs across the length of your back, right? And it is in continuation of the medulla oblongata. So they also this is also covered by durameter, arachnoid, and pyometer. The main thing is it has multiple nerves if emerging out of it at different points. So all these you will see. So they control all these nerves, which so they can directly relay the information to the nerves which are connected at the distinct region. And that is what we'll do in the peripheral nervous system. And this is exactly what it covers. So you have this spinal cord which gives out all these different kind of nerves at different region. So if you have a network of nerves in your thoracic region, in your cervical region, in your lumbar lower back, and then the sacral. And but there are, the main thing is there are two type of nerve fibers, one that gives the information and one that gives out the information. The first thing is efferent nerve fibers. Efferent means they are responsible for transmitting the message from the tissue to the CNS. So that means that is the one that will perceive the information, let's say the source of heat. Efferent are the one that will carry the information from the nervous system to the organ. Let's say the nerves that which runs to the hand, and then relate information is to take the hand up. So to efferent, you can think of efferent as something as coming out. So if it is efferent, it is coming out of some nervous system. Efferent is moving towards nervous system. And as I mentioned, they are further divided into somatic and autonomic nervous system. So somatic means some skeletal muscles so that you can control. Autonomic is the involuntary action, which is further divided into parasympathetic and sympathetic. Parasympathetic are basically those responses which are related to energy conservation, and sympathetic are what is body's way of dealing with any situation. So in that case, you will see dilation of pupils and inhibition of saliva. There is dilation of bronchia, increase in heart rate so that your muscles activities speed up and you are able to do whatever you. So these are generally involved in the flight and fight response. So you have two hormones, epinephrine and norepinephrine. Then work on this system. They first increase the sympathetic system so that you can do whatever you need. So let's say you have to run, so they will do all these increasing heart rate and then inhibition of digestive organs so that you carry out the response. And then you have the parasympathetic doing the exactly opposite, so that tries to bring everything back to its normalcy, bring back to these relaxed state. And so moving on to the unit components of nervous system, which is the nervous tissue. So if you see a nervous tissue, that generally consists of neurons, nerve cells, and the glial cells. And they are present in brain, in spinal cord, in the nerves. And what they generally do is they react. So they will receive, they are the ones that perceive the information and then relay to the brains and the spinal cord. And how do they do it? So this is a neuron in general. If you have to divide it broadly, it will be this cyton, cell body, and then the cell processes. So this cyton is mainly the like the uninucleus. So these are the nucleus and the cytoplasm. Cell processes are all these extensions from these cytons. So you have these dendrites, and then a long running node or long running chain, which is taking the information to the next part. So you have this multiple. So imagine these neurons being like in a series, and this is a dendrite that receives the information. It passes the information via this channel to this cleft. At this cleft you have a synapse, so will have acetylcholine or any neurotransmitter. The neurotransmitter moves to the next neurons, and the next neuron picks up the information again via their dendrite and move this on till it reaches to the brain or the spinal cord. And to see the structure, you have basically one nucleus. You will have these endoplasmic reticulum kind of structure, which is called Nissel's granules. You have these neurofibrils, which are responsible for the extension of these dendrites. Dendrites are what they collect the information. And then you have these axon, the main length which runs across the neurons. And then you have that these axons, they are protected or they're covered by Schwann cells or neurolemma. And wherever you have these myelinated sheets, so these are covered, these are myelinate sheets. If the myelin sheets are present, the small junction between them are called nodes of Ranvier. And why is this important? Because if so, if this happens, you will see when I say where it is present. So this is generally present in brain, where it is covered by myelin sheath. Now, when we do the conduction, we'll see that there is an action potential moving across the axon. If you have a myelinated sheet, these regions, they're imperceivable. So they cannot penetrate, or they cannot push the action potential. So what happens is the action potential moves from one node. So there's a jumping from these nodes. So these nodes of Ranviers are what gets the impulses. So the impulse move from here to here. So you see the information is relayed quite quickly, and that's why it is present in brain, because there the information is relayed very quickly because of the presence of these myelinated sheath. Otherwise, you have them long running chains. So it is moving via action potential all across the axons. Yeah. So that is what I just mentioned. So myelin sheath basically acts as an insulator, and they do not prevent leakage of ions. Okay. Now, based on what kind of where you have the dendrites and how many of them are present, you have these different kind of neurons that we just discussed. So you have sensory neurons. So they will pick these. You have these sensory neurons here. So they will pick the information for the stimuli and then pass it on to the brain and spinal cord. Then they're pseudo unipolar. So I had discussed this polarity of neurons in the original problem solving session. So this is based on from where your axon is derived. So you can see the axon is derived right somewhere in the middle. So it's in both directions. That's why it's pseudo unipolar, not just one axon running. Then you have motor neurons, which are these. Now, because they are motor neurons, again they have to be spreaded. So they have to have the multiple so that the effect is higher. So that's why they are multipolar. They have this multipolar nature, and their most important function is to transmit the information to the muscles. And then you have interneurons, which are between the sensory and motor, connecting them so that they can pass on information. Okay. Now coming to nerve impulse, because we have already done action potential once, it's easier to understand here. So nervous tissue also have the property like muscle to get excited and to conduct the impulse further. And we'll see that how it happens. So this is at the resting potential. Now this is your, so inside is called exoplasm, outside is called exolemma, the fluid that fills up your nerve fibers. Now these are ionic form. Basically, they have ions. So now let's consider first the outside environment. In outside environment, you have chloride ions, which are neutralizing the sodium ions. So sodium are positively charged ions. So in the outside, so basically here you have the sodium ions being neutralized by the chloride ions, and in the inside you have other negatively charged proteins which are neutralizing the potassium. So you have more of potassium, like potassium is present inside and more of sodium outside, right? So the membrane is therefore negative inside because you're like, we'll see why is this change, why is there this uh, polarity even at the resting stage. And that resting state polarity is somewhere around 70 to 90 millivolts, and it has an associated sodium potassium pump, and that is where the regulation of the influx and efflux takes place. Now uh, so the main purpose of nerve fiber or nerve muscle is to push sodium out and get keep sodium potassium inside. So potassium moves inside, sodium moves outside, right? Yeah. Now, why is there a resting state charge? It is because the permeability of the axon or the permeability of the layer is different for these two ions. So the potassium has more permeability. So what happens is there is more of potassium which is like lost. So the potassium keeps moving outside. So you should have had potassium inside and sodium outside, but because a positively charged potassium keeps moving outside, that's why you have more of a negative charge inside and positive charge outside. So that is why resting potential you have a negative charge. Good. Okay, now there is an impulse that is to be conducted in this nerve fiber. So the pump that comes to work here is again sodium potassium pump. Again, the sodium, which is supposed to be outside, is moved inside cells. So this is where your depolarization starts. Your sodium starts moving inside, and calcium was anyway, potassium was anyway moving outside. But sodium is now starting to flow in. As soon as your sodium starts flowing in, the first thing it does is to neutralize the negative charge. So there is already a negative charge, right? More of negative ions. The first sodium will come and neutralize this negative charge, and that happens somewhere around 30 millivolts, right? After this, when the sodium keeps coming in for more, then it leads to the action potential, which is this change in the polarity, and this keeps propagating all across the length of the axon, so the axon fiber, till it reaches to the end and it receives the information, it passes the information to the next neurons. So that's, and when this is happening, when it is moving, the portion which just got depolarized will go back to its normal state. It's called the repolarization. Again, the same thing will happen here, where now the potassium will be like pushed inside and sodium will be pushed outside, right? And again, this is mediated by acetylcholine and calcium 2 plus, as we have just read in the previous section. Okay, so again I have two slides on the questions for these topics. The first is, Which are the neurons? So this is what we have discussed already. Which are the neurons that carry impulses away from the central nervous, so which is coming out from central nervous system? Which of this has higher permeability in resting nerve state? We just discussed it, which is more permeable and keeps moving out of the fiber even at resting stage. The space between the arachnoid and the durometer is called what? I had mentioned this as well. And this also we have discussed. Myelin sheath comes from which cells? Yeah. So carrying away from central nervous system means coming out of central nervous system. So the nerves are called efferent nerves. Highest permeability was potassium. The space between arachnoid and durometer or durameter is called subdural. And Schwann cells are what myelin sheath are derived from. Next, so the action potential, okay, while the propagation of a nerve impulse is due to, so what is the movement, what is the actual movement of ions that is happening because of the which the action potential is propagating? Next, so a nerve impulse jumps from which region to another during saltatory conduction? So this refers to the myelin sheath. So when you have myelin sheath, it's called saltatory because it is jumping movement. So where does this jump actually takes place? Next is, what is the ability of neurons to initiate a nerve impulse call? So when does it actually, what is the feature called? And Broca's areas is related to which of these region in the cerebrum? Yeah. So the first one is sodium because it is sodium movement that initiates the action potential from the extracellular fluid to the intracellular fluid. Next is nodes of nerve. Ranvier, we did this. This is between the myelin sheaths. There is where the jump takes place. So the ability to get irritated or excited, because myelination is not related, because myelination is on the contrary does not lead to the conduction. Conductivity is the continuation of the action potential. Neurotransmission is at the end. The first step is the irritability or the excitability, and the Broca's area is related to speech. Okay. Again, the next topic, which again I will do in very brief, homeostasis, which refers to the endocrine system. Again, in very brief, so homeostasis is basically self-regulating processes where you have these different internal variables. They are maintained at a certain level. So basically, what it means is when there is a change in the environment, how your body keeps up with the environment so that your internal system remains at a stable condition. So you have these different variables, for example, body temperature, fluids, blood sugar. All these are different variables which are kept at control by this body, and this whole process of keeping them at a steady state is called homeostasis. So you have these different methods. So you have, for example, hypothalamus, as we will discuss in a few moments. They are responsible for controlling the temperature and osmotic pressure. Osmotic pressure means fluids in the body. Then you have evaporation, which you generally do when you sweat. You have this blood which are evaporated, because so when does sweating actually happen? When you start feeling too hot, and that is your body's evaporation is your body's way of coming down to normal temperature, cooling you down. Then you have pancreas and kidneys, which regulate blood sugar level and water balance. And blood is again responsible for distributing the heat. So your blood is distributing the heat throughout the body. You have this blood. So you generally see if there is increased blood movement, you feel heat. So that's blood's general idea. So like blood is responsible for distributing heat. And then you have the skeletal muscle, which is contracting and relaxing because of the very same reason. And other example of these homeostasis is when you have meals, your blood sugar level rises. Now, there is a certain extent to which blood sugar can be maintained. So your body's response is, okay, you have had sugar. Now we will utilize this sugar for all the energy that you can. So what starts is pancreas starts to give out insulin. Insulin says to the cells of the body, take up this glucose, whatever new has come into being. So these liver, so may all the glucose are finally stored in liver. So the liver, all the glucose are transported to liver. Liver stores it as glycogen, and because all the glucose has been taken off the blood sugar level or blood glucose level comes to normalcy, and that is a process of homeostasis. And this happens at several other layers because of several other hormones. And mainly, how does this take place? So you have a receptor in the same way that your like neurons are functioning, that there is an information which is sent to it, and then the information is relayed to the message to the center, and then there is an efferent or effector which is carrying out the information. In a very similar way, you have this homeostasis mechanism taking place. There you have a receptor who's perceiving a change in the environment. Then you have a control center, which is saying that, okay, this is the information, this is the output, and then sends the information to effector. Effector says that, okay, if there is, let's say, very crude example, body temperature is too high, the receptor receives it, gives information to control center. The idea is to get the body temperature down. So the control center will send information to the effector, which is the body muscles, saying that start evaporating, get your sweat muscles activated. You start sweating, your body temperature goes down. So that's the general flow of information. And this is perpetuated by these feedback systems, where you have negative feedback system and positive feedback system. Negative feedback system functions to get the variable, like you oppose the effect of the change. Basically, if you have blood temperature increasing, the negative feedback system gets activated, goes through the cooling mode, and it brings your body back to normalcy. On the contrary, again, if your body goes to decrease, like if body temperature goes down, then you have shivering. Shivering is a mechanism to bring your body temperature up, and then it goes back to normalcy. So any cue which is received, the body functions in the opposite way, so that the cue's effect is reversed. So that is called negative feedback system. And to perceive this, there are some set points, basically some ideal value for any variable, and any change from that normal value leads to this kind of feedback system. On the opposite to this, you have positive feedback. So mainly homeostasis is maintained by a negative feedback system, but you have these positive feedback system in some cases where they will work to enhance the effect of the change. This is, for example, in childbirth. So in childbirth, as soon as the baby moves or baby is about to come out, the information is relayed to the hypothalamus. Hypothalamus starts releasing oxytocin. Oxytocin starts increasing the muscle contraction, because of which you get in this pain are induced in the lady, and the mother is about to deliver. And the hormone, the oxytocin, induces further production of oxytocin, so that the contraction gets even higher. So this is all to enhance the effect of the change, so that to enhance the process of childbirth. So these are few examples of positive feedback systems. So you have these two feedback system working to maintain the homeostasis. And while talking only, I have mentioned that the main relay center for the homeostasis is the endocrine system, and the information output is in the form of hormone. So you have these glands which secrete this hormone, which carry the information, and they go and bind to their respective receptor, and they give the information out. So you have these endocrine cells, they release the hormone, they're released into the bloodstream, so they are ductless basically. And then they carry all throughout the body, go and bind to their respective place and desired effect. So you can go and bind to the skeletal muscle, or you can go and bind to any neurons to propagate the information. And these hormones, so they are released by glands, and there are several glands which are part of the endocrine system. It starts with hypothalamus in the brain, which has two regions: anterior pituitary gland and posterior pituitary gland. Posterior pituitary gland is mainly responsible for the secretion of oxytocin and ADH, only two hormones. One of them works on kidney, the other works on the uterus and the mammary glands in females. And the anterior pituitary has the major burden of secreting all the hormones which are controlling all the other endocrine glands, which involve thyroid, adrenal cortex, mammary glands, bones, and we'll come to that. So when we're talking about hormones and receptor, they have two kind of receptors. One is fixed membrane. So fixed membrane means that it will bind to the membrane only, and then there are this series of small molecules which will relay the information. So let's say there is an hormone which is supposed which brings the information of production or for initiating something. And if it binds to here, then you have these small molecules. At the end, it is a CAM, cyclic AMP. So this gives the information to the respective organelles that so and so activity is supposed. So let's say that hormone wants more. What or the activity of the hormone is to trigger sarcoplasmic reticulum to release more of calcium. So when the hormone comes and binds here, the information is relayed to the sarcoplasmic reticulum or the endoplasmic reticulum to release more of calcium. But in contrast to this, there are also mobile receptor hormones where the hormone move inside the cell and the receptor is somewhere in the cell, and it is generally in the DNA in the nucleus that goes and binds to the nucleus and initiates gene expression. So the ones which have mobile receptor, they generally are involved in enhancing a certain expression of a gene. And these hormones can also function as messengers. So it was coming here to like enhance gene expression, or in the other case to give out information. It can itself work as messengers. For example, you have hypothalamus, which is releasing something called thyroid releasing hormone, which is working on the anterior pituitary to release the TSH, which is thyroid stimulating hormone, which goes to thyroid. Thyroid releases two kinds of hormones, T3 and T4. Now, how does they act as a messenger? The moment T3 hypothalamus, and they give information to stop the production of the hormone any further. So that's how they control the feedback system as well. Now, talking about endocrine glands, these are the different endocrine glands, and let's start from the top. So you have the hypothalamus as the main center, which has the pituitary gland. So let's the function as well. So hypothalamus mainly controls the temperature, heart, and blood pressure. Pituitary, as we just saw that they have multiple function. Pineal gland that we were referring to when we were discussing about brain, they are also responsible in the regulating body sleep wake cycle. And how? By melanin secretion. So when it is dark, the pineal gland starts secreting melanin, which induces sleep. And when it is light, this production of the melanin stops, and because of which there's more of wakefulness. Next is thyroid, which is somewhere just below your throat. You have these two hormones T3 and T4, which regulate the body's energy, metabolism, and thyroid concentration. And working in conjunction with the thyroid, you have parathyroid. They also work for calcium absorption. Then you have thymus, which are responsible for the secretion of T cells. And then you have pancreas, which are responsible for the production of insulin, glucagon, the main hormone which are responsible for controlling the blood sugar level, and adrenal glands, which are responsible for cortisol and adrenaline, which are body's hormones which react to stress. And we will see that they are also responsible for the control of the excretory system. And then you have the reproductive organs, which are responsible in the respective reproductive systems health. So just to give you an overview, how does hypothalamus actually control all the other endocrine glands? So you have these hypothalamus, uh, posterior pituitary is called neurohypophysis, anterior is called adenohypophysis. And you can see that the different hormones that the different regions of hypothalamus releases, it affects different organs in different ways to release their respective hormone. And then there is negative feedback. So all these are under the control of feedback mechanism. As and when the concentration goes up, there's a feedback to stop the initiation or production. This is to keep the body in control. Okay. So again, there are some questions which tries to summarize this topic. The first is which hormones or which of these hormones are responsible for the flight and fight response. Next is pancreas is a mixed gland. So basically, pancreas is a ductless gland as well as a duct gland. So I'll come to this when I have given you a hint to answer this. So maybe you can answer from this. If not, I can discuss later. Then this is not a function of insulin and glucagon. So I'll give you first the overview of what insulin and glucagon does. So glucagon and insulin both are released by the pancreas in relation to blood glucose level. Glucagon, the idea of insulin is to reduce blood glucose level. Part of this we did in the first slide. Glucagon wants the opposite. Glucagon wants more of glucose in the blood, glucose in blood. So insulin will initiate several processes that reduce the blood glucose level. How can that happen? By storing glycogen, like storing glucose as glycogen, breaking up the glucose into simpler components and taking up into different tissue organs. On the contrary, if glucagon wants to initiate or wants to increase glucose, it will try to get glucose from different sources. It will break the glycogen, it will synthesize new glucose, it will give all the cues to increase glucose level in the blood. So they are working in antagonistic way to maintain the hemostasis. Now with this information, you have these two questions. Try to answer this. If not, I'll discuss it when we are discussing the answers. Yeah. The first one is epinephrine and non-epinephrine, or adrenaline and non-adrenaline. We had discussed this. Second is pancreas, the mixed gland, in which enzymes and hormones are secreted by separate cells. So hormones, while discussing, I had told that they are ductless. They are released directly into the bloodstream. Enzymes are rather secreted into ducts. They are secreted into the pancreatic and hepatic, so liver, pancreatic duct. So mixed gland is when it has both of these compounds being secreted, but they are by separate cells. So that's why the route of delivery is different. Now, which of these is not a function of insulin? So first is in decreasing glycogenolysis, decrease in the production of glycogen. What is glycogen? Stored glucose. So it decreases the production of stored glucose so that there is more glycogenolysis. Sorry, so glycogenolysis is breaking up of glycogen. So decrease in this breaking up of glycogen so that there is no glucose, and insulin wants to reduce glucose. Similarly, you have lipogenesis. So using up of glucose is different from glycogenesis is a glycogen synthesis. So it promotes glycogen synthesis. Gluconeogenesis means to secrete new glucose, and insulin tries to cater to lowering the level of glucose. So gluconeogenesis is something that insulin doesn't do. And the last is glucagon here. So glucagon does what? So it is related to glucose. The first answer is not correct. Second is it slows down glucose formation from lactic acid. This is not what glucagon does. Glucagon wants to accelerate the process. The second says decreases the conversion of glycogen to glucose. Again, not something that glucagon wants to do. On the contrary, it accelerates the process of glycogen breaking to glucose, because glucose level has to go up under the message or under the direction of glucagon. Okay, next set of questions are which of these hormones are not secreted by hypothalamus? Next is again, so it's a repetition of the previous question. Glucagon does what? Next is action of parathyroid, which is secreted by parathyroid gland, does what? And given us some hormones, their source and function, which of these is actually a correct match? Okay, a cue to remember the hormones for hypothalamus and not the pituitary gland is the presence of RH, or releasing hormone. So the first is the prolactin releasing hormone. Second is the cortex releasing hormone, thyroid releasing hormone. Cortex means adrenal. So releasing hormones are secreted by hypothalamus. FSH, SH is stimulating hormone. These are generally released by the anterior pituitary or posterior pituitary, generally by anterior pituitary. So the question was which is not secreted by hypothalamus, and FSH is the correct answer. Next is action of parathyroid. As we were discussing, it is pertaining to calcium level responses, so it increases the blood calcium level. Next is which is the correct match, which is the second option. Why is the first wrong? Because we will see that when we do vasopressin in the next part, which is the excretory system, vasopressin is released from the posterior pituitary, yes, but it does not increase the loss of water, rather it decreases a lot of loss of water. It induces absorption of water. Next is prolactin, which is said to be released by posterior pituitary and regulates the growth of mammary glands. So the function is correct, but the source is not posterior, because posterior only secretes vasopressin and oxytocin. Again, glucagon, it is from islets of Langerhans beta cells, and it stimulates glycogenolysis, which is again wrong, because uh, yeah. So the norepinephrine increases heart rate and is from the adrenal medulla. Glucagon, I'm sorry, the answer for glucagon is the alpha cells, because function is correct, the source is wrong. Okay. Okay, so last section for today's discussion and for this course problem solving session is excretory system. So excretory system is basically the system which is involving in releasing or excreting, pushing out the waste from the body. Now, it is generally in liquid form and solid form. So the liquid form is basically under high control of the I mean, the fluid which is in form of urine is highly under regulated control. So, a very general way of understanding is, so let's say there's a fish, freshwater fish. In general, freshwater fishes, they live in water less concentrated than the body fluid. So because they're freshwater, the water which is outside the fish has less concentration. Concentration means the solutes, concentration of solutes. So they're less concentrated than body fluids. So in general, we are talking about diffusion. We talk about movement across concentration gradient. Now here, fish therefore tend to lose salt because the outside system has less salt, fish has more salt, so fish body tries to move out salt. And because on the opposite, if solute is more, that means solvent is less, and solvent is less, so more, and solute is less. So because solute is less, solvent is more, so the fish tends to gain water and as compared to its environment. So in the fish, it does not have to drink water because water is moving in via the diffusion, and salt is taken in by active transport, because by concentration gradient salt will keep moving out. So its urine is basically large volume, but it is less concentrated than the body fluids, because it is continuously taking water in. So if the urine doesn't have any such concentration, like the solute is less. Compared to marine fishes, marine system or marine environment has more of salts and less of water. So the exact opposite happens in these kind of fishes, where you have the fishes, they tend to lose water because their water, with outside water concentration is less, so they will lose water, but they will gain salt. So that's why they have to drink water, because by like by their physiology, their water is moving out, so they need to take water in, and because salt is coming inside, they have to actively push salt out. So in their urine, first of all, they have less volume because they have to preserve water, because otherwise water is anyway being out, and the urine is slightly less concentrated than the body fluids. Now compare this to terrestrial vertebrate or humans here. Now humans tend to lose body water to air by evaporation, and we drink water to maintain the water hemostasis. And we also take in salt by voluntary movement. So our urine, on the contrast, is a moderate volume, and but rather of more concentrated value than the body fluids, because we work we in our body, the system works to have more body fluids, and it absorbs water at a frequent rate. Now, in this urine, what kind of products are given out is again based on what kind of organism you are. You could be excreting ammonia, you could be excreting urea, or you could be excreting uric acid. If you are an organism, basically if you are fishes and you excrete ammonia, they are called ammonotelic organism. If you are mammals, for example, amphibians, sharks, and bony fishes, some of them, and you excrete urea, you are called ureotelic. And if you are reptiles or insects or land snails, then and you are secreting uric acid, then it's called uricotelic. Okay. Now, for excretion, there are specialized organs in different organisms. For example, nephridia in our swans, flames have in planaria. Cockroaches have malphizian tubules. Prawns have these antennal glands in the frontal portion. Nematodes have something called rennet cells. And then you have also simpler structures like contractile vacuole, which is just a vacuole from which you also take in food and also give out food, which is present in the amoeba and the paramecium, again, which release ammonia. Hydra secrete or excrete their product by simple diffusion by general body surface. And then you have the tania and leucon. Leucon being the sponges, they also do by body surfaces. And then you have worms who have protonephridia, and the ferritima and paleomon, which have special glands. Moving to human excretory system, the main onus of excretion in humans is on the kidney. So kidney have multiple very broadly defined structures and intricately connected with nervous tissues to actually carry out the whole system. So you have these two kidneys here. If you take one of these in cross section and zoom it out, you see that they are consisting of these medulla and cortex. So cortex being the outer region and medulla being the inner region. Now you have these different. So if you pick one of these outside and zoom it out, and you see each of these have these kind of structures, which are called. So this is the portion of cortex, and this is the medulla. So the inner region and the outer region, and then you have a nephron, which is interspersed with blood capillaries, and each of these next nephron will have the glomerulus like structure, and all these want to do is to filter out the waste from the coming blood, pass it as urine, and reabsorb material which are or yeah, like pass the information, like retain the minerals or nutrients which are important to the body, and remaining they are passed on as urine. So the main steps of urine formation by these nephrons are the glomerular filtration, reabsorption, and tubular secretion. Is exactly what I just mentioned in very brief. So glomerular filtration is the, so if you see here, in each nephrons you have this structure here called Bowman's capsule and multiple glomerulus structure, which are basically capillaries. So what happens is that you have these blood capillaries moving in via that, right? So here the excess fluid and waste from products from the kidney are filtered out of the blood and into the urine collection tube. So if you see here, these are arterioles. These are the blood which have carried all the nutrients and the fluids. So they are taken from the fluid, the blood is filtered, and everything is passed on to the nephrons. How quickly this happens and how this determines how concentrated, so this is filtrate, right? So this is called glomerular filtration rate, and how, what is the pressure of this, like how many concentrated solutes it has taken, what kind of fluid it has taken, determines the fluid pressure rate. What fluid pressure? Filtrate pressure. So what exactly happens in this step is you have water and solutes which are smaller than proteins, and they are passed to move inside the capillary walls and pores of the renal tubule. Then you will see they have a very integrated structure. We will see here. So in these tubular structures, you have reabsorption of all the important nutrients like sodium, glucose, amino acids, and water. All these are reabsorbed and pushed into the blood again because they are important for the body. And whatever is not reabsorbed, they are released and they're secreted. And then there are some more components which are supposed to not be in the body, they're high. So you are secreting them into the blood, urine. So you have secretion of these hydrogen, creatinine, drugs. These are released into the urine and they're removed from the blood. So you have secretion and reabsorption as well. Finally make up the urine. So we'll see the step, how does it actually takes place. So first of all, we'll see the structure of nephrons. So nephron has these Bowman's capsule which are interspersed with these glomerulus, which has these capillaries. Then next to it you have the proximal tubule. Then you have this loop of Henle. The first part is the descending loop of Henle and then the ascending loop of Henle. And then you have the distal convoluted tube. So this is called proximal convoluted tubule because they are highly convoluted in structure, PCT. And this part is called distal convoluted tubule or DCT. And after all these, you have the final collecting duct which goes and connects to the renal pelvis, to the ureter and the uterus. So what happens is when you have the blood is being filtered out and all the fluids have been taken into the tubule. At the first step, you have the reabsorption. So this blue shows secretion and the pink shows reabsorption. So you have, so first of all, let's do the filtrate composition. What are filtered from the blood? Water, salt, bicarbonate, hydrogen, urea, the waste product, glucose, because that's present in the blood, amino acid, drugs, all of that the blood has taken from different cells of the body. It goes to the kidney and it gives it all out, except for oxygen, carbon dioxide, which just takes to the lungs and filters out there. Okay. Now in the filtrate, then the filtrate moves to the proximal tubule. You have reabsorption of nutrients, which are all these glucose and amino acids, salt, water, and bicarbonate. At the same time, kidney also is involved in purification. So it will secrete some drugs and the poison that it has filtered, detoxified into the urine and along with some hydrogen ions. Next, this fluid will now move into the loop of Henle. First, the descending part. So the descending part, the reabsorption of water takes place. So the descending part is permeable to water. So what happens here when water is moving out, the urine is becoming concentrated. This then moves to the ascending part. Ascending part is impermeable to water. So water is not no more moving inside. It has got concentrated and salts are also moving out. So because the salts are moving out, the concentration gradient is again reducing. So water is urine is again getting diluted because you are removing solute from the urine. And then you have the distal tubule. Again, water is reabsorbed. Again, it is getting concentrated. Bicarbonate is removed and salt is removed. And you are adding more of the potassium and the ions. And at the end, when it is going through the collecting tube also, if required, it undergoes reabsorption of salt, urea, and water as per need. So ultimately, the urine that is passed is more or less concentrated and of moderate volume. Again, as I was mentioning previously, this system is also under regulation by several hormones. Two of them are aldosterone and ADH. First, we'll see what aldosterone does. So aldosterone is here. But for the aldosterone to work, you have other checkpoints that needs to be passed so that your action actually takes place. So that is called angiotensin system. So renin angiotensin system is called RAS system. So the first control point for excretory system is the RAS system. Here angiotensin converts to angiotensin 1 under the presence of renin. Angiotensin 1 gets converted to angiotensin 2. Now angiotensin 2 has several effects. First is it causes the arteries to constrict, resulting in increase in blood pressure. Reduces the glomerular filtration rate, resulting in water retention and increased thirst. So what it is trying to do? It is trying to reduce urine and it is trying to get more of water in the body. Other than that, it induces ADH secretion. ADH is the vasopressin. ADH full form is antidiuretic hormone. Where it is secreted from? From the hypothalamus posterior pituitary. And it also stimulates aldosterone secretion from the adrenal cortex of the adrenal glands. So you have aldosterone and ADH. What do they do? So the aldosterone, it causes the nephron distal tubule to reabsorb more Na plus and water because the idea is to have more of water in the body. So it starts reabsorption at a higher rate, which is increasing the blood volume because it's going into the blood. And the ADH, what it will do, it mediates insertion of aquaporins into nephrons, collecting ducts. As a result, more water is reabsorbed. So water is absorbed, and remember that's why the previous answer was wrong, where it showed that vasopressin leads to loss of water. As we can see, that water is being absorbed by the body more under the influence of ADH. Similarly, they're also increasing the blood pressure. So you see that is the controlled by the hormones, and this renin is secreted by kidney in response to slow water. If there is less of water, kidney produces renin, and that starts the whole this process so that the water is increased. Similarly, you have ADH, which is not really doesn't have to be under the influence of renin. It can be directly secreted by the pituitary glands based on the water content of the body. So let's go over this. So you have water content of the this is the normal level. Now you have had too much of water to be drunk. So what the body wants the water to be pushed out from the blood or from the body. So it goes to the pituitary, and the pituitary now regulates the secretion of ADH. Now the ADH or vasopressin or antidiuretic hormone secretion is lower. What happens? There is low volume of water being reabsorbed, so the reabsorption volume goes down, and that's why you have very high diluted urine that is passed, and now you have low volume of water passing into the blood. So because most of the water is being excreted as urine, the blood volume goes down and the water content goes back to normal. Now opposite of this, you have eaten too much of salt or you have had too much of sweating, because of which your water level goes down. So now your body wants to increase the level of water. So it stimulates the pituitary to release lots of ADH, and we just saw what ADH does. ADH just increasing the reabsorption of water. So it leads to higher amount of water and the water goes back to normal level. Additionally, here you will also have thirst. Your thirst receptors will be activated and you will feel thirsty, so you will also drink more water. So you see, everything in the body is connected by somehow or the other, and there are different checkpoints for each of these to work. Okay, so the last two slides for today's session, and where we'll discuss questions on this topic. Okay, the first is ammonia is secreted by which of these following? So ammonia, first you need to know who secretes ammonia and what are the organs that are present in those organisms. Second is, so you have slight distortion in the right kidney and the left kidney in humans. Can you guess why? Next is the network of tiny blood vessels located at the beginning of nephron, be digit. And the last is malfeasant tubules are found in which of these organisms? Yeah. So the first one is gills, is because ammonia is present in fishes. Second is you have a considerable space occupied by liver on the right side. So you do know that liver and kidney are like kidney is just below liver, and because liver is carrying most of the space in the right side, that's why your right kidney is lower than the left kidney. The network of blood vessels which are present at the beginning of nephron is called glomerulus because Bowman's capsule is the encapsulation, and transfusion tubules are present in insects generally. And the last set of questions are this pressure facilitates filtration while urine is being formed. So, which is the pressure that actually facilitates the filtration? How does the filtration actually take place? This happens. So the next question is, this happens if the proximal convoluted tubule is removed. So what was the proximal convoluted tubule doing, and if you remove that, what could happen? Next is what produces urea as the excretory substance in the human body, of which organ is involved. And last is capillary hydrostatic pressure during filtration is built in the glomerulus as a result of what? So if you don't understand capillary hydrostatic pressure, it means capillary means blood vessels. So wherever blood vessels is involved, so where is blood vessel involved in the Bowman's capsule? Yeah. So the first answer is yeah. So which of this pressure facilitates filtration while urine is being formed? It is the filtrate hydrostatic pressure because so that concentration gradient is because of the filtrate hydrostatic pressure. If you have less, if you remove convoluted tubule, proximal convoluted tubule, your urine becomes more diluted because there is no absorption of water. And liver is the main gland which produces urea, right, because of a product detoxification. And capillary hydrostatic pressure is because of the afferent new arteriole. Efferent means the one which moves out towards the blood, being narrow than the afferent. So your because the movement of the blood gets restricted at the glomerulus, it gives rise to the capillary hydrostatic pressure. Okay, so that wraps up today's session and the session for this course in general. But if you have any doubt before your exams, then you are always welcome to ask in any of the videos or any of the discussion forums.